Hello, hello. Welcome back to What Is It Biology? I'm your host as always, Mr. Johnny Hopkins, and today we're going to be covering more random hodgepodge genetic vocab stuff. It all makes sense in the end, but you know, this is just another little thing. The next one, I promise you, it'll be out of this world. Well, in this world, because it's biology. But so it's going to be a little short little thing, as you can probably see, and you can probably see in the thumbnail. But so genetic vocab, we're going to talk. DNA is not all in the nucleus, and you know, a little bit about kind of evolution stuff. So, genetics. Fun fact: DNA is primarily in the nucleus in eukaryotes. You know, eukaryotes like humans, like me. I don't care about me, so I don't want to learn about these bacteria with DNA. I don't want to care about me with DNA. And not all my DNA is inside the nucleus. Some of it exists in the mitochondria. And if I were to say via plants, like those guys over there that you can't see, they actually have DNA also in the mitochondria, but as well as in their chloroplast. But because I really care about me only, we're gonna focus on these mitochondrial DNA. So what is the purpose? Like, how did this happen? Why is there DNA inside this fancy mitochondria? And the answer is endosymbiosis. Endosymbiosis is essentially this idea of how the mitochondria came to be. What we had was one bacteria and say hi to another bacteria, and then what happened is that this bacteria got eaten by the other one, so we had one bacteria inside another bacteria. And then that bacteria happened to be really, really good at just making ATP. And because as some of the bacteria have two membranes, they have one membrane inside, and then got the periplasm between, and then you got the outer membrane. And so what happened there is that this one was just really, really good at making ATP, and then bam, that guy just kind of happened to exist with DNA inside it. So what is the deal with these little circle guys, these little DNA? And so these are actually quite small. They're only about 16 kilobases. They're circular DNA, and they have no introns. So there's about 30 of these mitochondrial DNA circles inside the cell, and the abbreviation is mtDNA. So what does this little circle encode? It encodes 2 rRNA, the small and the large. Then you have 22 tRNA and 13 proteins in the electron transport chain, the ETC. And so what happens is that these guys are used to make those guys. Now, one thing to note real quick is that this is not the entire protein transport chain. It's not just 13 proteins. There's a lot and 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 a lot more than just those 13 proteins. So they actually get recruit proteins from other sources, which we'll get into a long ways down the road. But so, if you remember correctly, there's, what, 64 potential tRNA segments, but in the human cells and whatnot, there's usually a lot less because you have different ways of addressing this with wobble pairs, as well as the sliding in of adenosine over adenine. So, one thing to just quickly note is that genetic diseases and the way that genetics pass on between these mitochondrial DNA is that this is always inherited from the mother, because a sperm does not have really anything that injects in besides its DNA. It only injects in its DNA, and the mitochondria are always produced from the mother. So blame them on blame your mother's genetics on your lack of ability to make a lot of energy. So, and your addiction to coffee. So then we have genetic drift is potential. So these mitochondria have different DNA segments, and these DNA segments are not always the same. And so eventually some of them can be more populated than others, and then you kind of go down the line with progeny and progeny, and this random distribution becomes a little bit becomes even more random, and then you can have actually genetic drift where different proteins have different specialized functions, and we go on and go on. Fancy stuff happens, and we go to different species for evolution. Talk for a minute. Mutant mitochondrial DNA will also accumulate at old age. There's a lot less proofreading in the DNA polymerases in these guys, so you have a lot more mutants in old age. There's a little bit of fancy stuff that happens in the zygotes and non-somatic cells that we'll get into later that helps decrease this for passing it on to your further generations. So chloroplast DNA, though, because we can't just care about myself, we also have to care about my plants, my little pets over there. And so they have, they're actually about 120 to 160 kilobases long with just a linear structure, and they're encode 120 to 135 genes, with 76 of them being proteins and the rest being RNA. Like the 2 are RNA and just a bunch of tRNA to help, make the proteins that are encoded right there. So now we're going to go on to evolution a little bit. So blast. You're going to first call it blast. You just put some sequence in and you blast it. So BLAST is Basic Local Alignment Search Tool. It's used for multiple sequence alignments, MSA. And this is a very, very powerful technique for evolution as well as determining protein structure and protein function. So it's a very bioinformatic tool that, so essentially what you do is you insert a sequence of a DNA or a protein, and then it'll output just essentially a bunch of different species with very, very similar DNA and proteins sequences that are quite similar to it, just have like random point mutations or different little sequences, motifs kind of scattered about that are not the same. But if it walks like a DNA binding protein, it talks like a DNA binding protein, and it has a sequence like a DNA binding protein, that protein that you just inserted is probably a DNA binding protein. That's the general idea, trying to determine function and structure from these guys. So what we have with this is we have evolution. So essentially, paralogous sequences, we have diverging from the same gene. So we have one gene that gets copied, and there's 2 of them. So what happens is essentially they go into different types of functions. Very quickly, you can just imagine one protein could initially bind to DNA and say, figure out do some stuff with it, but along the way, another copy of it eventually just binds to RNA and becomes an RNP complex. It helps in the RNP complex. So quite easy to see how that could be a potential possibility for paralogous evolution. Then orthologous evolution, that are sequences that arise by speciation. Essentially, they have the same function, but they come from different routes. So they have different sequences and have a different way of doing things, but they still achieve the same function because they just didn't exist. But then magically now humans need to eat applesauce, and somehow a cat also needs to eat applesauce all the time, and they both develop these proteins that help digest applesauce. Something like that. So one thing to quickly note is that the number of genes is not comparable to the biological complexity of the organism. One really, really quick way to just say this is alternative splicing is a very distinct possibility, in that you could say they have a bacteria with 50 genes, but then if you have a yeast with 10 genes, but all those 10 genes can be alternately spliced 100 different ways each, then you've got something that's very, very complex. Whereas the bacteria has more genes, but the yeast is more complex just by the number of actually produced proteins and different biological functions that can be performed. That is obviously an exaggeration, as you can tell. So, DNA that is not closely related as well with between species and genes, typically they don't differ a lot by like 1 to 2% of that sequence. But so that's why this BLAST technique is actually very useful and very easy to use and understand where it comes from. Although there are a lot more fancy ways that it's able to pull things around. They're not just, oh, let's just kind of look at these letters and just kind of do a little basic match. It's a bit more complex, but that's a very, very easy way of just kind of thinking about it. As well as variations usually are in the gene copy number, large relations and duplications of the genes, and that can actually account for about 12% of the genome in most species. So that is genetic vocab. Essentially have DNA in your mitochondria. The DNA encodes RNA, tRNA, and some proteins in the electron transport chain so that it's able to make ATP. And then you have mutations and diseases that can be caused from these sort of things. And then you have MSA and evolution, that genes can essentially go to different functions or come to the same function with different sequences, and you're able to look at this using some bioinformatic techniques. So next time we'll be talking about the actual chromosome structure inside the cell and actually talk about some real story with histones and euchromatin and all that fancy stuff. So we'll see you next time. Thank you.